The quantified expression data is called expression index. And as we can see from the previous section, there are traditional approaches which measures RPKM or FPKM. This basically normalized by the library sequencing depth first before normalizing by the gene length. And earlier methods such as top hat and cufflink uses this approach. It's kind of a biologically intuitive approach. But later on, algorithms such as RSEM uses TPM or transcript per million. In this case, the RSEM normalized by the gene length first before normalizing by the scaling factor or the library size. And so the advantage of this approach is that the proportion of reads that are mapped to a gene in each sample is comparable, so that you can actually compare the expression index of a gene across different samples. So TPM nowadays is considered the more accurate approach to reflect the expression level of the gene in that sample. Later on, when we try to calculate differential expression between the conditions for a particular gene, we still use the count matrix or the count per million data for that calculation. And so the current best quantification approach is RSEM. You can either input a FASTQ data, which will take a very long time because it also needs to map, or RSEM can run on the BAM files to calculate the expression index. And it also takes a reference transcript annotation file. You can either include a gene level readout or a transcript level readout, or you can include just the coding genes, and some also include the non-coding transcript. So depending on how you want to annotate the gene, it will give a different quantification. The output is the transcript level expression, which include the read count. It can include the TPM, FPKM that's calculated on the effective transcript length. So you might notice that this is not the full gene length. It only considers the exons on the gene or the actual mRNA transcript, but it's not the full gene length. And you can see here, if we look at the read coverage across the gene, 0 means the 5' end, the beginning of the gene, and 100 means the 3' end of the gene. You can see here, most of the transcript has good coverage in the middle of the transcript, but towards the end, both for RT-PCR and also for sequencing, it's harder to reach the end. So the effective length is roughly estimated as the length of the transcript minus the average fragment length of the sequencing library plus 1. So that kind of reduce the reduce the length of the gene by this sequencing length. So you get a more accurate reflection of the expected reads on that gene. And we mentioned that depending on the reference transcript that we provide to the RSEM algorithm, it will try to estimate a different expression level. For example, if you only give it all the genes or all the exons without worrying about the different transcript or isoform, it can give you a gene level expression estimate. But if, say, this gene has different isoforms, which you can see use the different subsets of the transcript, RSEM will try to look at, the length on exon 1, 2, and 3, and look at the reads that's mapped to here, and use maximum likelihood to estimate the best expression level X1, 2, 3 to match the isoform structure and also the read that's landed in each exon to match this up. I won't go to the algorithm details, but I want to show you just kind of an example. Say this gene has 3 different transcripts, and we measure RNA-seq in the read landing in the 3 samples. Blue and red indicates the reads that's mapped to the plus or minus end of the read. And so this one is not a strand-specific RNA-seq. And so you can see here, because the first transcript is unique in having these beginning exons, therefore any reads that lands in here can help us estimate the expression index of that first transcript. And you can see here that is highest in liver, second highest in brain, and very low in muscle. And this part of the transcript is unique for the second transcript. So therefore, when there are a lot of reads mapped in here, this can be used to estimate the second transcript, which is, you can see, a very, very high level. For the third transcript, the exon is always shared by either the first transcript or the second transcript. But for example, because in the brain and the liver, the read coverage in here are potentially similar or even higher than the later half. Therefore, that essentially made the third transcript eliminates the likelihood that this third transcript is actually expressed. That's why you can see the isoform 3 expression index is estimated at 0. Whereas because we do have some reads in the later ones that are potentially higher, and those might be coming from the third isoform in the second sample in here. Therefore, even though the muscle has highest expression of isoform 2, it still have some residual expression in the isoform 3. And so you can see, depending on the transcript annotation file you provide to the algorithm, it will try to estimate a different transcript abundance. And so occasionally, the maximum likelihood may not really be able to give you the best, well, it will give you the best estimate to fit the data, but it may not really match perfectly. And usually that is because our knowledge of the existing transcript may not be complete. Maybe there's a hidden transcript that we didn't know, and it's contributing reads that is not explainable with the current annotation. So as we mentioned, RSEM is currently considered the most accurate, best quantification approach, but this runs quite slowly. You can either start from the raw FASTQ file or the BAM file, but that would mean you have to start running the STAR and then the RSEM to get the quantification. This is both CPU and memory intensive. Instead, there are pseudo-alignment algorithms such as Callisto and Salmon. These two are kind of competing algorithms. They actually have quite similar performance. And so instead of mapping the reads across the whole genome, because the coding transcript only accounts for about 2% of the genome covered regions. Therefore, if we just use the existing knowledge of the annotated transcript, we can just generate a mapping index out of the known transcript. And so this is only about, say, 2% of the genome. Therefore, the read mapping can be much, much faster. Yeah, so we don't really care about the exact, base pair alignment or some single base mutations. In this case, we just care about rough estimates of the expression level of the transcript. And so this needs the reference transcript annotation files. So we only give estimation of the known transcript. So basically it can, Callisto and Salmon can quickly map the reads into the known transcript and generate expression index file. Salmon has additional feature to correct for sequence specific and GC biases. This could arise when you run one sequencing machine or a different sequencing machine, or one day of the run or another day of the run. There might be slight GC biases, making reads with different GC content having higher or lower read counts, so which can be normalized across all the transcript. And so Callisto and Salmon again can run on both FASTQ and BAM files, but the real advantage is if you run it on the FASTQ, which will obviously be much faster. We can usually map tens of millions of reads only in a few minutes time. In homework 1, you will see that the Salmon estimated expression index and the RSEM estimated expression index are highly correlated, but they are not exactly the same. And so we sacrifice the accuracy with the improved speed and memory efficiency. This is an example of a Callisto output versus Salmon output. And this is on the same input file. And you can see here, the first column is the transcript, the transcript ID, and this is the original transcript length, and this is the effective length. You can see the two algorithms have slightly different calculations. This is the estimated count per million. In Salmon, this is called read number or number of reads. It's kind of a count value. And then this is the TPM value estimated, and that's kind of the final expression index that we want to use. If you look at these measurements, you can see that they are, fairly similar. 0.12 here is 0.13, next one is 0, and this is a bigger one, 0.63, 0.73, and so on. So overall, Salmon and Callisto give quite similar results. Even though they are not quite as accurate as RSEM, it's much, much faster and efficient.